Introduction, and I would like to thank the organizers, and I'm particularly honored to be giving this lecture. So, as you know, I'll be speaking to you about the use of pluripotent stem cells in regenerative medicine. And as you're all very well aware, there are two major hurdles in regenerative medicine, namely the shortage of tissue and the problem of immune rejection. And so with the advent of pluripotent stem cells, we now have the opportunity, hopefully, to solve both of those problems. So, as you may be aware, there are two main ways to make patient-specific cells. The first, of course, is somatic cell nuclear transfer. The concept there is very simple. You start out with an empty egg. You take the cell from the patient you want to make your stem cells from, put it next to the egg, fuse it, create your pre-implantation embryo, and then you end up with, eventually in the laboratory with stem cells that match the nuclear donor cell. The other approach, of course, is cell reprogramming, where you upregulate the transcription factors, and then you end up with iPS cells. Again, that can be done for specific patients. So, as you know, human SENT isn't quite there yet. No stem cell lines have been produced, at least not that are diploid. We and other groups have made advanced stage embryos, and we've actually shown that they do, in fact, reprogram the nuclear donor genome back to a state of embryonic state comparable to an embryo. We've shown that the markers of pluripotency, NANOG, OCT4, etc., are all upregulated. And if you look at the global gene profile, here are the nuclear donor cells to begin with. After reprogramming, they're down here, right next to normal human IVF embryos. By contrast, if you try to reprogram the human genome using rabbits or cows, there's virtually no reprogramming that occurs. So just to show you the power of this technology, and in a large animal with a sophisticated immune system similar to a human, we actually studied these old cows, equivalent to humans, probably about 70 years old. We took a skin biopsy, and because we were going to create cloned cells, we had to knock in a neo-cassette so that when we put them back in, we could discern the clones from the normal. And what we actually saw was that these cells were very youthful, so whether you're talking about iPS cells or SENT or whatever, you have an upregulation of telomerase. So without any myeloablation or any drugs, these cells, once they were injected, were able to take a home through competitive repopulation. Actually, we estimate anywhere from 5 to 10 times more competitive than the adult cells. And we saw the neomycin cassette show up in the first 12 weeks. And if we took a sample of the peripheral blood at 6 months and looked at it, 60% of the colony-forming cells were of clonal origin, and they generated these large white cell colonies, which you only usually see in youthful animals. So, again, the goal here wasn't just to give an old cow a new immune system, but in humans there are over 100 autoimmune diseases, lupus, multiple sclerosis, etc. So the idea is that hopefully we can use these pluripotent stem cells to treat some of these disorders. This is another study we did with Piero and Aversa. This is a cross-section of a mouse that had a severe myocardial infarct. This is the healthy tissue. This is the infarcted tissue. And again, we used cloned hematopoietic stem cells, this time laxity positive. And within 1 month, you can see here in blue, they infiltrated the entire infarct, and we saw about 40% of the damaged myocardium replaced. So we also can make pluripotent cells, of course, from iPS cells. The advantage there, of course, is that in addition to having the ability to create virtually an unlimited supply of cells, and just so you know, for all of our clinical trials, we started with 1 embryonic stem cell, and that would be enough to begin, once we've made our master cell bank, to treat everyone on the planet. So these are immortalized cells, and that really does come into play for therapeutic purposes. But more importantly, for the controversy, there are no eggs and no embryos involved. Of course, we're still trying to perfect the reprogramming techniques, not only for safety, but also we published, I think it was in the early 2010, to show that there were problems with the major two methodologies, whether it was the Yamanaka or the Thompson methods. Those methods led to apoptosis, limited expansion, and early senescence. And just to show you an example of what was going on here is if you looked at a range of different embryonic stem cells here and compared them to both lentiviral-derived iPS and retroviral, you could get a feel for this, because we were mainly interested in translating this into the clinic. So here you see these hemangioblastic intermediates that go through multiple cycles so that we could create erythroid cells. And they're very robust colonies, and we were able to get very large amounts of these erythroid progenitors. By contrast with the iPS cells, very small colonies. In fact, there was a 1,000-fold difference in the ability of the iPS-derived cells, and we studied almost a dozen of these lines compared to embryonic stem cells. Now, since that time, since 2007, there have been new methods that do not integrate into the genome. So here's a paper we published with Dr. Kim at Harvard where we actually use direct delivery of proteins. But of course, there's also now the episomal methodologies, the mRNAs. And it looks now, at least in our hands, with the right reprogramming factors, we're now able to pretty much solve those problems. So it looks like we're almost there, if not there. Let's see what the FDA has to say about that. But until that is perfected and we have regulatory approval to proceed, you're going to see the first clinical trials, probably mainly focusing in the central nervous system and the eye. A few reasons for that. One is, of course, they're immune privileged, so without the patient-specific cells, you can go in with off-the-shelf allogeneic cells. Two is, of course, there are major diseases that afflict those different organs, whether in the brain for Parkinson's or Alzheimer's, but also in the eye, for instance, of course, age-related macular degeneration. And then thirdly, as anyone who works with embryonic stem cells knows, neuroectoderm is almost a default pathway, so we can get very high purified numbers of these different cell types very easily. So we're focused in on the eye. We're studying multiple different cell types, hemangioblastic cells for ischemic retinopathy. We're working on corneal endothelium for corneal disease, MSCs for uveitis and autoimmune diseases. We also have some cells that are for glaucoma, and also we're focusing mainly right now on the use of retinal cells for the treatment of these retinal degenerative diseases. So I'd like to talk a little bit now about our clinical program. We got approval from the FDA to begin clinical trials. This was back at the end of 2010, using what's known as retinal pigment epithelium, or RPE cells, to treat age-related macular degeneration, but also Stargardt's disease, which is one of the leading causes of juvenile blindness. And of course, AMD alone affects up to over 30 million people worldwide. It's the leading cause of blindness in the developing countries. And the degeneration of these RPE cells is a critical player in the cause of that disease, as well as Stargardt's and literally a dozen other diseases such as retinitis pigmentosa. So there have been numerous animal studies showing that these RPE cells can rescue photoreceptor function and attenuate visual loss. So we think that pluripotent stem cells are ideal for creating healthy, youthful RPE cells for transplantation into patients. So this is just a cartoon of the eye, just to show you what's going on. The light enters into the eye, it hits the photoreceptors, again, the cones and the rods we see with, and underneath those are the RPE cells, which they basically maintain the health of that environment. So they provide critical nutrients, they're involved with vitamin A metabolism, and they also phagocytose the shed photoreceptor fragments, and they absorb stray light and protect against UV light. So again, there are numerous advantages using pluripotent stem cells. First of all, again, you can make a virtually unlimited supply of these cells. I mean, we can, from 1 6-well plate, make enough cells, at least for this disease, to treat hundreds of patients. We can also make them under GMP conditions, completely pathogen-free. We can minimize batch-to-batch variation, and we can thoroughly characterize them to ensure their optimal performance. And as regards RPE, we have studied dozens of lines, and virtually every line makes these RPE very robustly so they can be passaged, characterized, and expanded. So if anyone has grown ES cells in the Petri dish, and you leave them there long enough, virtually all of those will eventually develop these little pigmented patches, these little freckles, and you can mechanically remove those, dissociate them, and it turns out that they will then de-differentiate, expand to confluence, and then once they reach confluence, they'll reestablish the pigmented cobblestone morphology, and they'll go through this differentiation, de-differentiation again and again and again. So from a very tiny number of these cells, you can create billions and billions of these cells. So we've characterized these cells extensively. They're more or less identical to primary RPE. We show a downregulation of the markers of pluripotency, an upregulation of the RPE-specific markers. Again, one of the critical features of these cells is that they phagocytose the shed photoreceptor fragments. So if you throw these latex beads into the culture, they'll eat them, and then you can quantitate that using flow cytometry. So, great in a petri dish, what do they do in animals? So we first looked at the Royal College of Surgeons rat, the RCS rat. We injected these human RPEs subretinally, and this is an animal that has macular degeneration very similar to humans. And we did a dose escalation study, and we showed 100% improvement in visual acuity over the untreated controls. Similarly, we studied a Stargardt's mouse model and showed that we could prevent the onset of that disease process. And so if you look at these animals at any point during the course of the disease after implantation, you can look at the human anti-human markers, and then this is bestrophin, an RPE-specific marker, and show that they form beautiful monolayers and integrate nicely in the eye. So in the RCS rat, after several months, naturally, if you look at the outer nucleated layer, the photoreceptor, there's only one scraggly layer of these cells up. These animals are essentially blind. When you transplant these RPE cells in, this outer nucleated layer is very robust, 5 to 7 layers thick. So that was what we were hoping we might see happen in humans. So for regulatory approval, we had to go through and show that this process is not permissive to undifferentiated embryonic stem cells. So we go through the entire process, and we're actually have developed an assay where we can detect even one undifferentiated pluripotent stem cell in over 1 million cells. So our maximal dose that we're going to be using is 200,000 cells. So it's quite sensitive. We can actually go in towards the end of this process and actually spike that with up to 10% undifferentiated cells, and we still don't end up with any undifferentiated cells at the end. We've done extensive safety studies. We've shown that there are no safety signals, no tumors, ectopic tissue in over 200 animals that we studied. We even spiked some of these animals. The RPE was spiked with up to 1% undifferentiated cells and still no teratomas. So based on that data, we got FDA approval, and at this point we've studied 18 patients, a few more being done in the next few days. And so again, as I mentioned, we have 2 clinical trials in the U.S. One of those is for AMD, and there are 12 patients in that study, and another one for Stargardt's. And we also got approval to do the first pluripotent stem cell trial in Europe as well, and those are actually being done at Mooresfield Eye Hospital in Scotland. We also are doing these clinical trials in the U.S. at 4 sites here. They're all in the top 5 eye institutes in the country: the Jules Stein at UCLA, Wills Eye, Vascular Palmer, and Mass Eye and Ear. We treated the first 2 patients back in July of 2011. And so this just shows you the clinical trial design. And so we started with the first cohort of patients, 3 patients at 50,000, and then we go up to 100, 150, and 200. So we start out with 1 patient in each group, give it some time, and after we get approval from the Data Safety Monitoring Board, we do the next 2 patients, and this repeats again for each dosage. We're over halfway at this point. We recently got approval from the FDA now to add on some patients with better vision. So the initial patients were legally blind, so they had a vision of about 20/400 at best. And now we have permission that we'll be starting very soon with patients that have only 20/100. So the goal of this therapy isn't in theory to reverse blindness, but it's to prevent the onset. So once the RPE die and the photoreceptors die, that's irreversible. So our goal is to get in there earlier before you lose your photoreceptors. So we published some of the early work early last year on the first 2 patients. And this was published in the Lanza. And so this just shows you some of the results. So this is the injection site. Pre-implantation, there's no pigmentation. And then you see after 6 weeks and 3 months an increase in the pigmentation. Actually, using OCT, we can actually look in real time into the eye and see what's going on almost at the cellular level. So here you can see the RPE attachment to Bruch's membrane. So looking at the results, this is the first Stargardt's patient. This is the untreated eye. So this patient could only detect hand motion, and throughout the course of the study, could only detect hand motion. By contrast, in the treated eye, what we saw is within 1 week, that patient was able to start counting fingers. Within 2 weeks, the patient could start counting letters on the visual acuity test. And by 1 month, she could actually read 5 letters on the visual acuity test. That was at 3 months. We're continuing to follow these patients. This patient is now out over 18 months. This is a progressive disease. The vision should be getting worse. Instead, it's continuing to get better. But that doesn't really capture the difference this is actually making in the quality of life in these patients. So these patients now can actually, a lot of them are saying they can see colors and blues and all these different, they can read their watch, they can go to the mall on their own, they can go shopping. So it really is impacting them. They all want it in the other eye. So this is an untreatable disease at present. So the hope is perhaps as we go into the better vision patients, we may see even more dramatic results. So this is another patient just to show you that these cells are indeed engrafting. So these are allogeneic cells going into the eye. So this is 1 day post-implantation. So this is the injection site. You don't see anything. 2 months. Here you see the papules of RPE growing in half a year. Very significant. So these are sheets of RPE that are not rejecting. And of course, there's no immunosuppression at that point. So we have other programs that we're hoping to translate into the clinic as well. We have a corneal repair program. So there are over 10 million people that have corneal blindness. It's the most transplanted organ. At one point, they used to transplant the full thickness of the cornea, but now it's more popular just to transplant the thin layer of corneal endothelium on Descemet's membrane, and we can now make entire sheets of these corneal endothelium. This is an interesting project we're also working on, because I thought it would be interesting just to make this point, is these are retinal neural progenitors. They're actually photoreceptor progenitors, and so we were looking at this, to replace photoreceptors. And we made this unexpected discovery, and that is that first you inject these systemically, and if you look at what's going on in the eye, it turns out that there's something called an ERG, which is the same as an EKG, only in the eye. And so you can measure the A wave, which is for the cones and the rods, and it turns out that after 2 months, spectacular results in the treated animals compared to the PBS. And so what's going on? So if you do histology on the eye, the untreated animals, this is the outer nucleated layer, the photoreceptors, almost completely gone. The treated animal, here, very robust layer of outer nucleated layer is quantifies it. So you say, oh, this is great, so what? But what we found, interestingly, is we went back into the eyes, did PCR, you could see the mouse primer, no human cells in the eye. But if you think about it, that makes perfect sense. You inject these systemically, and they're getting pulled out, probably all in the lungs. So they're not even going to the eye, yet they're rescuing, in a very major way, the vision and the outer nucleated cells. So what's interesting in this system is that the cells we use do not have that biological effect just a few weeks earlier. So we can look at these at the proteins using antibody arrays and identify those few molecules that are different, the few cytokines, and then hopefully create a cocktail. So learning from these cells what to do to help patients. Another program we're involved with are these so-called hemangioblasts. These are bipotential cells that in the embryo go on and become the vasculature, as well as the lymphoid and myeloid cells. So we figured out how to make these in very, very large numbers from human embryonic stem cells, as well as iPS cells, and you can label them with GMP, and we did a study with Maria Grant down at the University of Florida with ischemia reperfusion injury to the eye. We injected these into the TL vein. Within 24 to 48 hours, they had migrated to the damaged retinal vasculature, and you can see them starting to incorporate. By contrast to an eye uninjured, no incorporation. Very smart cells, they know where to go. In fact, if you inject them intravitreally, they'll just sit there as sheets if the eye is undamaged. This is another study that we did with Malcolm Moore's group at the Sloan Kettering. This is an ischemic limb model of a mouse. So this is the Doppler of the hind limbs. This limb, there's no blood flow to begin with. One injection of the hemangioblasts within a month, and we have complete restoration of the blood flow. Very robust collateral circulation. Similarly, in a severe myocardial infarct model, we're actually able to cut the mortality rate in half. This is a study we did actually in a spontaneously diabetic rat. Again, as you know, all the secondary complications of diabetes, most of them are microvascular in nature. So when we injected these cells into the animal, again, if you look into the eye, you can see the green. These are the hemangioblasts repairing that damaged vasculature. But you can imagine that anywhere that there is damaged tissue that are sending out the signals, these cells will probably go into that site to repair the damage. So I've spoken to you about what they can do in terms of vasculature. So again, if you put them in culture media that enhances their growth into that lineage, you know, you get these capillary-like structures, the uptake of LDL. But if you take the exact same cells and put them into culture media that favors hemangiogenic, or I should say, blood lineages, you can actually turn those into erythroid cells, myeloid cells, multilineage cells. And then we took advantage of that to turn them into entire tubes of red blood cells that actually uptake oxygen and just like normal transfusible blood, we were actually able to get them to enucleate tube because, of course, red blood cells are enucleated. So we've been able to make red blood cells, you know, from all the different blood types, whether it's A, B, O. In fact, we've now made IPS lines, and we even have an ES line, originally starting from an O negative donor, so we can make red blood cells that will match virtually everyone in this room. More recently, we've been focusing a lot of our attention on making megakaryocytes and platelets. Of course, as we know, there's a huge medical need for platelets. They're very short-lived. They only live a few days. So, you know, this is a serious problem. So the way platelets are made is you get these immature megakaryocytes. They fuse to make these larger multinucleated megakaryocytes that then go on to make what you can see, these proplatelets, and then they break off to make the platelets. And so, you know, in terms of doing this, there are many tricks, including microfluidics, that allows us to really increase the efficiency of that quite considerably. So we now know how to make these platelets very purified in very, very large numbers from both IPS and ES cells. It turns out these platelets undergo clot formation and retraction, just like normal platelets. We can show in a mouse model of laser-induced arterial injury that they will actually participate in the clot formation. And more recently, we've done studies using macrophage-depleted animals showing that these human platelets circulate like normal platelets. So this is a cross-section of some of these platelets. They're biconcave, like normal platelets, the same size, about 2 microns. And they have all the outer structural components as normal blood platelets, including the alpha and dense granules. So we studying these and carrying out all of the different assays that the regulatory agencies require. This is a spreading assay that shows that these cells will attach and spread like normal platelets. And so we actually met with the FDA a couple of weeks ago, and so the hope is to begin the first IPS trial, hopefully this year, using these platelets. But another feature here that's very, very important is we're also dealing with, for instance, BOTA or Homeland Security. If there was ever a nuclear weapon or a radiological weapon, and a lot of people were exposed and needed platelets, they're short-lived. There's just no way to get that many platelets. The beautiful thing with this technology is that you can freeze these progenitors down and have enough for, say, 1 million, and then within a few days have enough platelets for 1 million people in a few days. So it's really ideal. You just freeze them down until you need them in case there's ever some sort of emergency. So stay tuned for that. And finally, but also of great interest, is we have a program for mesenchymal stem cells. And as you all know, I mean, they can be made, obtained from bone marrow, adipose, even tooth buds. They differentiate into fat, bone, cartilage. They're immune privileged, which is particularly important for off the shelf. They migrate to the site of injury. They exert immunosuppressive effects. And at this point here, because of the great interest in these, there are over 200 clinical trials currently listed on the government site. So why use pluripotent stem cells instead of adult stem cells? Well, first, of course, is you can use these right off the shelf for immediate use. They're an unlimited source, basically. They're very easy to derive. They can be expanded in very large numbers in vitro. They turn out to be more youthful and live longer than the adult cells. And indeed, actually, anyone who's worked with MSCs knows that after you passage them, you can actually use those as negative controls because they lose their biological abilities. And so we're able to actually create very, very large numbers of these. So this is a little cartoon of Wally. If he represents the number of MSCs you can get from 1 unit of bone marrow, the same equivalent made from these ES cells could fill all of Fenway Park. So we can get over 30,000 times as many of these cells. But that's just numbers. So what did they do in terms of biological efficacy? So this is just one study that we did. This is in the multiple sclerosis mouse model. So in these untreated animals, they actually start out with a clinical score of about 2 to 4, which basically means that they have complete or at least partial paralysis of their hind and rear limbs. I can't show you this, that's been published yet, it's in process, but basically these animals move around, and if they do, they're dragging their hind limbs. By contrast, one injection of these ES-derived MSCs, the animals have no leg paralysis, jumping around, almost completely normal, score less than 1. By contrast, the bone marrow-derived MSCs had a small effect, but did not eliminate the paralysis. So again, this is very important because these cells obviously could be used for many, many applications. Again, all the autoimmune diseases, MS, osteoarthritis. We are currently studying these now in a model of chronic pain, stroke, spinal cord injury, long, long list. But for us to translate this into the clinic, the reason these are ideal is you don't need any immunosuppression. They persist transiently, and we can even irradiate them. So if we irradiated our MSCs and used them in the MS model, still knocked out the effect of the disease. So finally, to realize the full potential of this technology, we really need to use these pluripotent stem cells as a source to reconstitute more complex tissues and structures. Of course, that's the whole area of tissue engineering. So this is just a bladder that Tony Atala had made using tissue engineering techniques. And this is some work we did with Tony many years ago where we actually had these little miniature renal devices. So these are polycarbonate membranes. And in this study, we had 3 groups. One was an unseeded scaffold. This little bag is our primitive attempt at a bladder. And what we did is we seeded the polycarbonate membrane with either allogeneic renal cells that were, this was a cow model, so we wanted to test the immunity in a large animal. And these were cloned renal cells. So again, we put these back into the animals. In the case of the cloned animals, they came from the nuclear donor animal, so they were basically genetically matched. We placed them under the skin, and 3 months later we removed them. And sure enough, these actually connected up with the vasculature of the animals. The unseeded, of course, we didn't see anything in our primitive bladder. The allogeneic cells, they were reduced, destroyed immunologically. But the cloned cells had actually, you could see in each of these little, they're about the size of a postage stamp, you could see straw yellow material, which indeed was removing toxic material from the blood of the animals. They had elevated amounts of creatinine and urea. And if you looked at it histologically, you could actually see that the cells had assembled into glomeruli, into tubules, and that they actually appeared to connect up in a unidirectional fashion with the polycarbonate membrane to concentrate those materials. And then finally, of course, this whole area of pluripotent stem cells has been marred in controversy because embryonic stem cells generally require the destruction of an embryo. And so we published a couple of papers where we actually showed that doesn't need to be the case, that we actually exploited the advantage of using PGD, pre-implantation genetic diagnosis, where what you do is you just pluck one cell from an 8-cell stage embryo, and we can create entire embryonic stem cell lines, and then the biopsied embryo continues on to the blastocyst stage unharmed, it's frozen down, and the embryonic stem cells go on just like normal embryonic stem cells to make all 3 germ layers. They have all of the markers of pluripotency. Actually, because they're coming from the modular stage, we're finding in many cases that these lines are the lines of the best lines of any that we've studied because they seem to have a little extra pluripotency. I don't know if that's really the case. But in any case, we can now make these lines without any embryo destruction. And finally, I want to acknowledge the people who did all the work. That includes all the scientists at Advanced Cell Technology, and I particularly like to say thanks Steve Schwartz at UCLA and Tony Atala at Wake Forest.
questions if there are any.
What's the status of the blood, so the O negative blood, how far along are you with that?
So we can get very large numbers of these cells. The main bottleneck there is that we're getting primitive erythroid cells, so that they're nucleated. So our efficiency right now in nucleating is about 60%. And for moving that into the clinic, that isn't quite good enough. And so it's a major effort. We're still doing it, but we've shifted right now most of our resources into the platelets. But it definitely is doable, and they appear to transport oxygen and they react very much like normal transfusible blood.
That was actually what I was going to ask about. Is the problem really in the field has been getting cells to the adult cell stage? I mean, you're talking about their being youthful, that's great, but you normally need cells to be have adult capacity, functional capacity, and where else besides the erythroid lineage?
Well, even in the erythroid, if you think about it, obviously the embryo is still nucleated, and there are actually people who do just have the primitive hemoglobin, so they live quite fine. So that it is a possibility. But you are right, we do want to mature those into more to the
And have you found strategies in which to push cells, for example, like cardiomyocytes or anything like in that area towards more adult-like cells?
Oh yeah. I mean, our platelets are absolutely mature. All the organelles, all the components, all the particles are completely identical to normal platelets. And except for the erythroid lineage, almost all of the cells are virtually identical to the primary cell you would find in the body.
That was fabulous. If I understood it correctly, your 90 blast studies